Today I'm excited to talk to you about the fascinating topic of crop domestication, and I'll be covering the early days of domestication all the way to today, and then reflect on the future of domestication. So I want to start with why we're here today. We're here because of events that happened in our shared human history, when our civilization moved from hunter-gatherer societies to agricultural societies, which is also known as the Neolithic or Agricultural Revolution. A major driving force behind this revolution was crop domestication. This is when wild crops were consciously or subconsciously bred to make them more amenable to farming and to our diets. So this is just like the fascinating example I'm showing you on the slide right now that illustrates how corn, as we know it today, was bred over thousands of years from a plant known as teosinte that looks remarkably different. And actually, all the major crops we're cultivating today went through similar events that allowed them to acquire agriculture-friendly traits. So from one crop to the next, there were quite a bit of similarities in the traits that they acquired. And that's what's called the domestication syndrome. And they acquired things like larger fruits or grains, reduced branching to facilitate cultivation, loss or reduction of seed dispersal so that seeds don't shatter before you even get to harvest them, loss of seed dormancy, which allows seeds to germinate synchronously, changes in photoperiod sensitivity to expand the geographical range we can grow them in, and making them more nutritious while removing toxic compounds. And this happened through a process known as selection, which I'm going to simply illustrate here. So let's say that we have a bunch of wheat plants, and that one wheat plant had bigger seeds. So this one would get selected as the parent of the next progeny, and likewise, let's say that among the descendants, one plant produces more seeds, so this one would get selected, and so on and so on. But something was happening behind the scenes that was unbeknownst to the farmers until DNA was discovered in the last century. It's that each plant has its own genome, which was a total black box for thousands of years. But without knowing it, farmers were selecting genetic variants. So here represented one genetic variant, the one that results in bigger seeds with a red bar. And so after this genetic variant was selected, another one would get selected. So in this case, one that will lead to more seeds, and that is represented here by the addition of a yellow bar. But the issue with this blind walk and not knowing what their genomes are is that it takes a very long time. It relies on luck and random events like naturally occurring mutations, which are quite rare. Over the last century, things changed quite a lot. There was major progress in our understanding of DNA and how to start manipulating it. One tool that plant breeders got to use is mutagens to increase the number of genetic variants in a population. With mutagens, we're no longer at the mercy of mutations appearing naturally over a long period of time. We can quickly generate a lot more genetic variants so that we have a lot more to choose from to find desirable traits. We also saw the emergence of sequencing that allowed us to decipher these black boxes so that we can start understanding the underlying genetics behind crop traits. And then, of course, there was the advent of GMOs, where once we uncovered certain useful genes, we could introduce them into crops. So for example, if we identified a useful gene in wheat, the idea is that we could introduce it in, let's say, rice to obtain a similar trait. And so enter the new biotech era we're living through right now. And today we'll be highlighting two groundbreaking developments, CRISPR and advances in genomics, starting with CRISPR. The CRISPR revolution started a decade ago when Jennifer Doudna and Emmanuel Charpentier reported in the journal Science that the defense system from microbes known as CRISPR could be bioengineered and harnessed as a gene editing tool. And this is actually what later earned them the Nobel Prize in 2020. At its core, the CRISPR system is a defense weapon against viruses. On the slide here, you see these spider-like creatures on the surface of a bacterium. And these creatures are actually viruses known as phages that invade bacteria. So we have those viruses sitting on bacteria, and what they do is inject their own DNA inside as a way of hijacking bacteria to force them to become virus-producing factories. And of course, bacteria don't like that, and so they've developed a defense mechanism against it. And this is exactly what CRISPR is. They first take a stretch of DNA that got injected. Then they integrate it into their own DNA, and they will use this information they acquired to make a weapon. And that's CRISPR-Cas, which is effectively a molecular scissor that will cut the invasive material. and this is how bacteria can stave off infections. So this system was bioengineered to create a tool to modify DNA sequences of our choice. So here we have a DNA sequence with 4 colors representing each of the 4 base pairs, so the 4 different types of nucleotides, the 4 DNA letters. And what happens next is that a bioengineered guide RNA will invade the double-stranded DNA at a predefined location, and it also recruits the Cas protein to this DNA region that will then cut the DNA. So now we have the DNA of our choice that got cut, and the cell will go into what you could consider as panic mode to fix this break, and in doing so, it may make a mistake. So it could result in the nucleotide change, an insertion of a short stretch of DNA, or a deletion of a short stretch of DNA. And so what this means is that you can introduce mutations at specific DNA sequences, but the only problem with that is that you don't have control over the exact mutation that will be introduced. Today, we're moving away from that into what I would call CRISPR 2.0 that allows us to go beyond this panic mode I just described, and where we actually have full control over the mutations that are introduced. So for example, if we want to insert a specific predefined sequence, we're now starting to be able to do that. If we want to delete a predefined sequence, we're starting to be able to do that as well. And then finally, if we want to modify specific nucleotides into other ones, we're also starting to be able to do that. So now that we can rewrite and write in DNA, the main question is what sequence should we even write in or rewrite? And for that, advances in genomics have been crucial. To explain this, let's take rice as an example, because it was the first crop to have had its genome sequenced. Something that is really interesting to think about is that there is a huge number, about 780,000 rice varieties that are stored in gene banks around the world. As I just mentioned, rice was the first crop to be sequenced. It made the cover of Science in 2002 when the draft of the genome was released. But in reality, only 2 varieties of rice were actually sequenced at that time. And while it was a huge advance, there was an entire universe of rice genetic diversity that we were missing out on. But this is now changing for 2 main reasons. First, the costs of sequencing a genome have dramatically decreased over the last 2 decades, even beating Moore's law. The first human genome, for example, cost billions of dollars 20 years ago, but today we can do that for less than $1,000. Second, we've also seen major advances in long-read sequencing that are allowing us to assemble whole genomes more accurately. So on the right side of the slide here, I'm showing you the Oxford Nanopore, which is allowing us to do sequencing at the bench and directly plug it into our computer to get data. As a result, the concept of pangenomes has recently emerged. And so pangenomes mean that instead of focusing on one genome, we're sequencing multiple genomes from multiple varieties so that we can get access to much more genetic information. And so by looking at a population of genomes, we can better understand how genetic differences result in different traits. So in this slide here, I'm showing you very recent publications on rice pangenomes. And the point I'm making here is that pangenome studies have been proliferating over the last couple of years. And so while this slide here is for rice, this is happening with every other major crop. So to recap what I just said, we now have powerful genetic tools and we have access to a lot more genetic information so that we know what genes to manipulate for different purposes. And so we're starting to see headlines like these about the improvement of crops like tomato, wheat, rice, and corn. So for example, on the slide here, tomato was edited to enrich it with GABA, which is a health-promoting compound. Wheat was modified to resist fungal infections, and rice and maize had their yields boosted using CRISPR. But I would argue that there are even more exciting applications for CRISPR, and that's the extreme acceleration of domestication. For example, a study published in 2018 in Nature Biotechnology was able to use CRISPR to partially redomesticate the wild ancestor of tomato, Solanum pimpinellifolium, into plants that look like modern tomatoes. They did this by CRISPRing genes that are known to have played roles in domestication. By CRISPRing the fruit size gene, fasciated, they obtained bigger fruits. By CRISPRing the fruit number gene, multiflora, they obtained inflorescences with more flowers. And by CRISPRing the growth gene, self-pruning, they obtain plants that are more tame so that they can be more easily cultivated. So what this study suggested is that we can significantly accelerate the domestication of plants using CRISPR. And of course, in tomatoes, it was just a proof of concept since tomatoes are already domesticated, so we don't need to redomesticate them from scratch. But where this has the biggest potential is with orphan crops. These are crops that have some really interesting characteristics and that play important roles in limited geographies and local economies, but they have been quite underinvested in. And that's why they're called orphan. It's because they were pretty much neglected by modern breeding. But now CRISPR can change that. So here's an example. That's ground cherry, which some of you may be familiar with from farmers markets, for example. The issue is that this crop is not currently optimized for large-scale cultivation. And it's actually a great candidate for accelerated domestication using gene editing because it is closely related to tomatoes, so we can use the information that we acquired in tomatoes to facilitate ground cherry's domestication. And that's what a publication in Nature Plants reported. Researchers were able to semi-domesticate ground cherry by CRISPRing domestication genes. So by CRISPRing the growth gene self-pruning, they obtained, just like in tomatoes, more tamed plants. And by CRISPRing the fruit size gene clavata1, they obtained larger fruits. Another similar great proof of concept was conducted in a huge cousin of rice, Oryza alta, which is in the middle here. And for comparison, you can see on the right side of the slide, Oryza sativa, which is the domesticated rice we're used to. So same thing here, a research group reported in the journal Cell that they could semi-domesticate it as well. By CRISPRing the hormone regulation gene semidorf, they were able to obtain smaller plants, and by CRISPRing the seed morphology gene grain size 3, they obtained significantly larger rice grains. And so the main point to keep in mind with these examples is that gene editing is able to reduce the time it takes to domesticate plants from thousands of years to just a few years. And so you can now start to imagine the potential for a bunch of other crops like the orphan crops I just showed you. Now let's take a step back and look at the big picture. When we look at some of the major crops we're growing today, you could argue that there is actually nothing particularly special about the wild ancestors they were domesticated from. So as you can see in each one of these examples, wild ancestors of potatoes, of corn, of wheat, of cotton, of tomato, and of soy, they've come a very long way, from tiny fruits and grains to these farming-friendly and large crops that we have today. And in fact, we could even argue that it was partially a fluke that these crops and not others got domesticated. The map I'm showing you right now indicates where the crops I just showed you in the previous slide experienced their early domestication days. And so a way to think about it is that the wild ancestors of these crops happen to have been at the right place at the right time in human history, and that had things been slightly different, it could easily have been another crop that got domesticated. And yet today we're locked into a limited number of crops compared to the large diversity of other potential crops that are out there. And that's largely because of some complex factors. We have specialized infrastructure for certain crops, and we also have established supply chains for them. We have government incentives, and of course we have consumer habits for specific crops. And we still need to fund R&D to develop new crops. So there isn't an easy solution to unlock all of this for new crops. But as a society, we probably don't spend enough time thinking about domesticating your crops and what we need to do for them to be part of the future of agriculture. But I hope that today I convinced you that now is a great time to start doing so. And on that thought, I'd like to thank you for your attention.